Hi students, today I will go for the translation mechanism. So the translation is the basic mechanism we know when from the particular RNA protein have been generated. It's a mechanism of the translation. So translation is a much more formidable challenge in information transfer than the transcription of the DNA into the RNA, and unlike the complementary between the DNA template and the ribonucleotides of the messenger RNA, the side chains of the amino acids have little or no specific affinity for the purine and pyrimidine bases found in the RNA. So the machinery responsible for the translating the language of the messenger mRNAs, that is mRNAs, into the language of the proteins is composed of four primary components, that is mRNA, tRNA, amino acyl tRNA synthetase, very important, and the ribosome. You have to go through the functions of the ribosomes. Then comes to the messenger RNA or the mRNA. Here we will go for the term ORF. ORF means open reading frame, and which specifies a single protein and starts and ends its internal sites within the mRNA. So the translation starts at the 5' end of the open reading frame and proceeds one codon at the time to the 3' end. So 5' to 3' orientation in which the translation process can occur. So the first and the last codon of the ORF, known as, obviously we know that the start codon and the stop codon. So the start codon will obviously be the AUG or methionine, maybe it GUG or valine. Then comes to the part of the prokaryotic mRNAs have a ribosome binding site which recruits the translational machinery, and in this case we have to go through a particular sequence which is known as the Shine-Dalgarno sequence, very important. So what is this? This particular Shine-Dalgarno sequence to facilitate the binding of a ribosome, many prokaryotic open reading frames contain a short sequence upstream start codon known as the ribosome binding site or the RBS. This element is also referred to as the Shine-Dalgarno sequence, after the scientists who discovered it on the basis of comparing the sequences of the multiple mRNAs. So the extent of the complementarity and the spacing between the ribosome binding site and the start codon, that is start codon means either AUG or GUG. In maximum animals or maximum living beings, it will be AUG. It has the strong influence on how actively a particular open reading frame is translated. So the high complementarity and proper spacing promotes the active translation machinery. Here we will learn the translation machinery only in case of the prokaryotes. It's important, not in the eukaryotes. So next I will go through the structure of the tRNA. It's a clover leaf structure, very important, and there are some of the parts known as the acceptor stem, because it is so named because it is the site of the attachment of the amino acid is formed by pairing between the 5' end and 3' ends of the tRNA molecule. The 5' CCA 3' sequence at the extreme 3' end of the molecule protrudes from the double-stranded stem. The Chi-U loop is so named because of the characteristic presence of the unusual base Chi-U in the loop. The D loop makes its name from the characteristic presence of the dihydrouridines in the loop. It's an unusual loop. Generally, in case of the DNA or RNA, we never find this kind of the base. So next, come to the part of the anticodon loop. So it is, the name implies, contains the anticodon, a 3 nucleotide long decoding element which is responsible for the recognizing the codon by base pairing with the mRNA. So cloverleaf representation of the secondary structure of the tRNA. Here we will find the acceptor arm, the Chi-U loop, the variable loop, anticodon loop, which can code the anticodon, and next the D loop. Then comes of the tRNAs have the L-shaped 3-dimensional structure. I have given you the notes on that. Next comes to the part of the amino acyl tRNA synthetase charge tRNAs on the 2 steps. That is, the main thing is the tRNA charging, in which the adenylated amino acid which is composed of the adenyl group, so it is known as the adenylated amino acid, which remains tightly bound to the synthetase, reacts with the tRNA, and this reaction results in the transfer of the amino acid to the 3' end of the tRNA via the 2' or 3' hydroxyl and the concomitant release of the AMP. So there are two types of the RNA synthetase, tRNA synthetase enzymes, class 1 and class 2, which helps in this process mechanism. Next comes to the part of the each amino acid tRNA synthetase attaches a single amino acid to one or more tRNAs. And next comes to the part of the tRNA synthetase recognize unique structural features of the cognate tRNAs.